we're going to review cell membranes. Now, you've all covered cell membranes in biology and in microbiology and in anatomy, and we're going to do it again, but even go into more detail. This is going to be a very important subject for this physiology course. The cell membrane acts as a semi-permeable boundary, allowing some chemicals to go in or out of the cell, obviously limiting, though, which ones can get in, which ones can go out. At the molecular level, your cell membrane is made up of a double layer, which means, or bilayer, bi meaning two, double layer, of phospholipid molecules with embedded proteins. Let's see a picture of this by looking at the previous page. So this is the bottom of B20. I already colored mine in. You can color yours. You don't have to color. All right. Let me explain it. All right. Here's a cell. All right. This is the cell membrane. The fluid inside the cell is the cytoplasm. Cytoplasm is 80% water. We refer to this cytoplasmic fluid as ICF. Anybody remember what ICF means? Good. Intracellular fluid. You'd say, when did you tell us that? Page B1. If you don't believe me, look up page B1. And all living cells we learn on page B1 have to be surrounded by fluid or they'll die. This is called the ECF, the extracellular fluid on page B1, or tissue fluid. Now what we want to do is just to magnify or enlarge this area, because the cell membrane is the interface between the cytoplasmic fluid on the inside and the tissue fluid or extracellular fluid on the outside. So here, enlarged, is the cell membrane. There's the cell membrane. Now, the cell membrane is made up of a double layer of phospholipids, as we've already reviewed in section A. We know that each phospholipid kind of reminds me of a balloon with two strings. The balloon part is hydrophilic, and the strings, which are two fatty acids, are hydrophobic. So notice that the inner layer of phospholipids, the balloon part that likes water, faces the cytoplasmic fluid on the inside of the cell. And in the outer layer of phospholipids, the balloon part that likes water faces the tissue fluid. The tissue fluid on the outside of the cell. And what's in the middle is pure fat, all hydrophobic. And I've just said that I described that like an Oreo cookie, right? Like a cookie and a creamy filling in the middle. And that creamy filling of fat, since water and fat don't mix, right? That double layer of fat basically creates a partition or separation between the cytoplasmic fluid on the inside of the cell and the tissue fluid on the outside of the cell. That's the function of the double layer phospholipids. But more important to us are these embedded proteins. There are a whole bunch of different proteins embedded in the cell membrane. Some of these proteins are sticking out on the outer surface of the cell membrane. This is the outer surface. Some of these proteins are facing the cytoplasmic fluid on the inside of the cell. Incidentally, if I were to represent like these proteins on the outside, so that, in this picture, these would be sticking out on the outside of the cell membrane, if that makes sense. And some of these proteins are facing the cytoplasmic fluid on the inside of the cell. Some of these proteins extend the full thickness of the cell membrane. So we want to talk about what these different proteins do. So the first ones we're going to mention are these right here, these proteins that extend the full thickness of the cell membrane. In your picture, it's labeled a channel. And the purpose of the channel proteins, and this is like a cutaway view. A channel protein is like a pipe. It's like a tube. It's like hollow in the middle. And these tube-shaped proteins allow ions to flow in or out of the cell. They are ion channels. Now, we wrote this on B21. Let's look on B21. So on page B21, what are the functions of these proteins? So number one, some of them act as ion channels. Now we already know what ions are. Ions are electrically charged atoms, atoms that have a different number of electrons than protons. So there are sodium ion channels, and potassium ion channels, and chloride ion channels, and calcium ion channels. In other words, it's not like all these ions flow through the same ion channel. They are specific. They are specific. There are specific ion channels. So the sodium ion channels are only for sodiums to flow through. And the potassium ion channels are just for potassium ions to flow through. So they're very specific. Furthermore, these ion channels can open or close. Most of them, most ion channels are closed most of the time, but they can open. But they can open or close, these ion channels. Now, just to give you a context of how we're going to be learning more about this, some of you may have heard of a class of drugs. I can't remember if I've mentioned them before, I may have. There's a class of drugs known as calcium ion channel blockers. Has anybody ever heard that term? Calcium ion channel blockers. Sounds like drugs that would block the calcium ion channels. In fact, there are all kinds of drugs that affect these ion channels, as we'll be seeing. Okay, now, the next thing listed are transporter proteins, or carrier proteins. And I wrote that transporter proteins transport sugars or amino acids across the cell membrane. These are known as transporter or carrier proteins. Again, there are specific ones. So the ones that transport sugars are different than the ones that transport amino acids. Now you'd say, okay, I'm going to look for, hold on, Professor Fink, I'm still writing this down. I'll give you time to write that. Let's just look at our picture. On our picture on the previous page, so here it shows, somewhat arbitrarily, I'm pointing to what's labeled a transporter or carrier protein. Now here's the point. These ion channels are big enough to allow these tiny, tiny ions to go through. Remember, an ion, like a sodium ion, is an electrically charged atom. Glucose, a sugar like glucose, is 24 atoms big. You'd say, how do you get that? C6H12O6. That's 24 atoms. It is way too large to flow through an ion channel. So the only way that sugars can get across the cell membrane is they attach and they are helped across the cell membrane by these so-called transporter proteins. Similarly, there are different transporter proteins that help transport or move amino acids across the cell membrane. These and amino acids are even larger than can be larger than sugars. They can be. So these need help to get across the cell membrane. Now, we wrote on page B21 the terms active transport and passive transport, or facilitated diffusion. You'd say, yeah, like what do you mean by that? If the transporter protein requires ATP as a source of energy to move that, say, amino acid across the cell membrane, that's called active transport. But sometimes the transporter protein does not require ATP. It does not need energy to help move a molecule across. If no ATP is required, that's called passive transport or facilitated diffusion. Usually, as we'll be learning, usually the difference is that if the chemical that's being transported across is flowing in down its concentration gradient, it doesn't require energy. But if it's being forced against its concentration gradient, it does require energy. Furthermore, amino acids usually require active transport, but interestingly, sugars usually do not require active transport. Most sugars are transported by passive transport. Now, at this point, you might say, I don't know, I think I know what he's talking about, I'm not sure. Let's help show you another picture. First of all, look on the next page. On B22, Wow, what a great diet. Look at this. Isn't this terrific? On B22, it lists, we're going to be talking about what we just did what ion channel proteins are. We're talking about transporter proteins. We're going to be talking about receptor proteins and enzymes and cell identity markers. And look at this. This is a summary chart of all this stuff, and it's got pictures and everything. Let's look at one more picture. Take a look at page B26. B26. So on page B26, here it's showing the cell membrane, right? Diagrammatically, it's a double layer phospholipids with the proteins embedded in it. And it's showing these are proteins. And it's showing here a box-shaped chemical can attach to this protein. And this protein transports or carries that box-shaped chemical across the cell membrane. Because this particular protein requires ATP to move this particular chemical, that's called active transport. On the other hand, in this protein shown right below it, it shows some ball-shaped chemical attaching to a different protein. And at least in this case, no ATP is required for this carrier or transporter protein to help that ball-shaped chemical across. That's why that's called passive transport or facilitated diffusion. You'd say, well, Professor, I'm really glad you wrote that. Well, it's actually all written right here. It says specific solutes can bind to specific protein carriers which channel them through the membrane by carrier-mediated transport. The transport may be active, requiring metabolic energy from ATP, or passive, requiring no metabolic energy. So it actually says it right there. All right, so that's, we'll have more to say about these transport proteins. All right, so that's for moving sugars and amino acids, larger nutrients across the cell membrane. Now, the next protein we want to mention on the bottom of B21, on the bottom of B21, are enzymes. Of course, enzymes are arguably among the most important proteins in a living thing, including us. They are proteins that catalyze biochemical reactions. There are thousands and thousands of different enzymes to catalyze thousands of different chemical reactions. So, and again, on the picture that we had on the previous page, I just somewhat arbitrarily labeled this an enzyme. Okay, could the enzyme be also located on the outer surface? Could be. Could be on the inner surface of the cell membrane. So there are a lot of enzymes. Everybody got this? Okay. Now, on page B23, what's another role of proteins? Now, if you've had biology, you certainly learned about, so far what I've talked about, some of these proteins acting as enzymes, as ion channels, as transporter proteins. You may or may not have heard of, maybe, linker proteins. And I wrote that linker proteins affect the cytoskeleton and shape of the cell. So our first thought is, what does that mean? First of all, let's show you where the linker protein is. In our picture, back on B20, having you flip back and forth, but on our picture, the linker proteins are always located on the cytoplasm side of the cell membrane. That's where they are located. They're on the cytoplasm side. These linker proteins are attached to the cytoskeleton of the cell. Now you'd say, what's the cytoskeleton? You should have learned that term in both a biology class and anatomy class. You'd say, well, I did? Look, we have, over there, we have Mr. Skeleton Man. Our skeleton, right, kind of creates a structure for our body, right, so that our body kind of stays together. That's what holds it all together. Inside of a cell, there are proteins. And we know that the cytoplasm is mostly water and then proteins by weight. And these proteins are not just floating around in the cell. These proteins are organized into a complex pattern called a cytoskeleton. So this cytoskeleton is made up of an arrangement of proteins. It's made up of an arrangement of proteins inside the cytoplasm of the cell. These linker proteins are attached to the cytoskeleton proteins. And we're going to be seeing that these linker proteins, which are attached to the cytoskeleton proteins inside the cytoplasm, can cause these proteins, the cytoskeleton proteins, to move. And then when they do, that causes a change in the shape of the cell. So the way that certain cells are able to change shape, you'd say, yeah, like which cells change shape? White blood cells. White blood cells kind of crawl around the body like an amoeba, right? They exhibit cytoplasmic streaming. And the way they're able to move and change shape involves these linker proteins and movement of the cytoskeleton protein. Okay, and so I wrote on page B23. On B23, we wrote linker proteins affect the cytoskeleton and shape of the cell. Now, another very important protein, I would say among the most important of all the proteins, certainly in the cell membrane, are receptor site proteins. Now, these receptor site proteins are proteins that can be activated by hormones, neurotransmitters, and other chemicals. Any chemical that activates a receptor site is known as a signal molecule or ligand. You'd say, like, do we have to know those words? Yes. So know that any chemical that activates a receptor site is known as a signal molecule or a ligand chemical. A ligand is a term that's used in biochemistry classes as something that attaches to something else. Is a ligand. Now, let's learn a little bit more about these receptor sites. Let's look on the next page. On page B24, okay, on page B24, it shows what's labeled an endocrine cell. You'd say, what's that? That's a cell that secretes a hormone, right? We call the glands that secrete hormones endocrine glands. So, and a gland, endocrine glands, made up of endocrine cells. Now, maybe this is a beta cell of a pancreatic islet, islet of Langerhans that's secreting insulin. All right. Maybe it's a cell of the testes secreting testosterone. Maybe it's an alpha cell secreting glucagon. All right. So it's secreting a hormone into the bloodstream. All right, the definition of a hormone is a chemical carried in the bloodstream. Now, hormones and neurotransmitters and other chemicals, drugs, affect some cells but not other cells. Now, why would a hormone affect some cells but not other cells? Receptor sites. So here it shows a cell that does not have a receptor site for this hormone. So, therefore, even if the hormone diffuses out of the bloodstream, it has no place to attach to this cell, and so there's no response. On the other hand, here is a cell with a receptor site for this hormone. So if this hormone diffuses out of the bloodstream, it can attach and activate that receptor site, causing a response by that cell. So any cell that is activated by a particular chemical is called a target cell for that chemical. This is a target cell for this hormone. This is not a target cell. This cell is not affected by the hormone this cell is. So this pretty much explains why, again, hormones may affect some cells but not other cells of our body. Now, these receptor sites, again, are specific. We've talked about specific ion channels, specific carrier or transporter proteins. There are specific different enzymes, and there are specific receptor sites. So a cell might have one or two different receptor sites. It might have 30 different types of receptor sites. So it might have a receptor site for insulin, a receptor site for glucagon, a receptor site for dopamine, a receptor site for epinephrine, a receptor site for growth hormone, a receptor site for estrogen. That would be a cell that would be affected by many different chemicals. And we could have another cell that might only have receptor sites for 2 chemicals. So again, but each receptor site is specific for a specific hormone or neurotransmitter. There's always specificity, meaning it's always specific. Now, let's look at something else. Right here, just to the right. Now, we have talked previously about how there's 2 major categories of hormones. Remember that? There are protein hormones and steroid hormones. And we know that steroid hormones are actually made from a fat called cholesterol. And protein hormones are made out of, like all proteins, amino acids. And then we had previously learned that, you'd say, well, okay, like who cares? So we had learned that one important difference is that a steroid hormone can be taken orally, but a protein hormone cannot. Because just like cholesterol, any food containing cholesterol, that's absorbed right into your body. A steroid hormone, which structurally looks like cholesterol, is just absorbed right into your body. It's not digested or broken apart. So a woman can take estrogen in her birth control pills orally. But a protein hormone, any protein that you ingest, any protein, is digested into amino acids. So if a person were to swallow insulin, it would be the same effect as eating actin and myosin in meat and muscle cells. It's just going to be broken apart into amino acids. So we learned protein hormones have to be injected. Here's another difference. Since steroid hormones are made from a fat called cholesterol, they can easily diffuse across the fatty membrane. Because like dissolves like, a fat can move through that fatty membrane. So steroid hormones easily diffuse across the cell membrane so that the receptor sites for steroid hormones are inside the cell. They're either in the cytoplasm or more commonly they're in the nucleus. Now why is the receptor site for a steroid hormone in the nucleus? When the steroid hormone activates the receptor site in the nucleus, that initiates transcription of a gene. That's really what steroid hormones do. They cause a gene to be copied, that's called a messenger RNA, and then that's going to lead to the production of some protein. That's really how steroid hormones work. All right. Now, on the other hand, the most other hormones and neurotransmitters, the protein hormones, the neurotransmitters, they are not made from fats. They are not fat soluble. They cannot easily get across the fatty membrane. So the receptor sites for all the other hormones and neurotransmitters are on the surface of the cell membrane. Certainly proteins and protein hormones are way too big to get across a cell membrane. And even neurotransmitters, which are very small, are not fat soluble. They're not fatty like a steroid hormone made from a fat called cholesterol, so they can't get across the cell membrane. So these receptor sites are on the outer surface. So now we know that the location of the receptor sites are different for a steroid hormone versus a protein hormone or a neurotransmitter. Let's consider something else. Let's look on the next page, page B25. So on page B25, so on B25, here it's showing these receptor sites on the cell membrane. That's what we're focusing on right now. And it's showing a signal molecule, a ligand, a chemical. They call it a ligand. Signal molecule or ligand. Could be a hormone, could be a neurotransmitter, could be some other chemical. And it's attaching and activating this receptor site. And as a result, it shows we get a response. Right? You get a response. It activates it. Let's give you a specific example to have in mind. So, I think all of you have heard of adrenaline. Adrenaline is actually a registered brand name for epinephrine. So we use the words epinephrine and adrenaline interchangeably. All the books call it epinephrine, because if you say adrenaline,